In the apparent silence of the inner ear, there is a microscopic battle, the outcome of which can mark the fate of our relationship with sound. Listening is a very delicate biological act, a molecular choreography in which each protein must fulfill its role with almost poetic precision. When something fails, the echo that disappears never returns. If you like our content, do not forget to subscribe and give like, so you can be aware of the latest discoveries in the fascinating world of biology and hearing health. Your support is essential to continue sharing valuable information. An investigation presented at the 70th annual meeting of the Biophysical Society has revealed an unexpected mechanism that could explain why sensory cells in the ear die without the possibility of replacement. Scientists have studied ear proteins as simple electric channels, but this new finding suggests additional complexity. When these proteins, TMC1 and TMC2, become out of control, they can trigger irreversible death of hearing cells. This is not just a functional breakdown, it is an intimate structural collapse that redefines our understanding of deafness. In the depths of the inner ear, ciliated cells act as translators of the sound world. These cells convert mechanical vibrations into electrical impulses that the brain interprets as sound. This process, known as mechanotransduction, is crucial to our ability to listen. TMC1 and TMC2 proteins are fundamental in this process. Although they have been considered the nucleus of the channel that allows ion flow, there is an uncomfortable truth. When these cells die, they do not regenerate, which means that each cell lost is a definite silence. The finding of the National Institute on Deafness and Other Communication Disorders proposes that TMC1 and TMC2 also act as lipid scramblases. This means that in addition to being ion channels, they have a crucial function in the redistribution of lipids in the cell membrane. When membrane asymmetry breaks, a sign of apoptosis occurs. In models with mutations in TMC1, it was observed that the membrane loses its symmetry, leading to cell decay. Cell death is not only due to electrical failure, but to structural collapse. The new findings also address the ototoxicity of certain antibiotics, which were traditionally thought to block the function of TMC channels. However, they could trigger scramblase activity, causing structural damage to hearing cells. This discovery opens the door to designing antibiotics that maintain their effectiveness without activating the destructive function. In addition, the involvement of cholesterol in this activity suggests that metabolic factors may influence hearing vulnerability. We are faced with a paradigm shift. Proteins that allow hearing not only capture sound, but also guard the integrity of the cell membrane. When that custody fails, the price is irreversible.